Hello, my name is Jennifer Terry. I am a graduate student mentored by Dr. Lori Neuman-Lee, and today I will present endocrine and immune patterns in freshwater turtles at Bearitage Farms, Arkansas during the active season. All organisms must allocate finite amounts of energy to various processes through trade-offs. These trade-off dynamics are context-dependent on the individual and the environmental level. Glucocorticoids are expected mediators of these trade-offs. As an example, the innate immune system is then influenced by these endocrine patterns. Unlike birds and mammals, turtles and other reptiles must get their heat from an external source. Turtles have long lifespans and many reproductive opportunities. There's a disparity between males and females in terms of reproductive obligations, and turtles rely primarily on the innate immune system. For my project, I compared endocrine and immune responses to an acute stressor across demographic groups throughout the activity season. We captured red-eared sliders for 7 to 10 days each month between May and October using baited traps. We collected blood samples, morphometrics, sonograms, and then used this plasma collected from the blood to perform bacterial killing assays and radioimmunoassays to assess innate immune function and corticosterone concentrations. I then conducted Kruskal-Wallis and post-hoc Dunn's tests using Program R. Bacterial killing ability, or a measure of the innate immune function, varied in females across months. In particular, there were lower levels of killing ability between May and July compared with August and October. There was no significant difference between bacterial killing ability from samples collected at the 30 and 60-minute time points. When grouped by sex, bacterial killing ability was lower in juveniles than adults, as shown in Part A of Figure 5. Circulating corticosterone concentrations, however, were higher in males than they were in females and juveniles, as indicated in Part B of Figure 5. It is also important to note that females had relatively no change in circulating corticosterone concentrations throughout the acute stressor. In conclusion, the response to an acute stressor varies by sex and time. Males may have access to more energy to mount an appropriate response to stressors than females and juveniles. Juveniles are likely still developing their immune system and also are diverting more energy towards growth. Females are likely mobilizing a lot of energy toward clutch development and nesting in May through June, are then deprived of energy by the end of the nesting season in July, and then regain energy throughout the late summer into fall through foraging. This may explain why we saw higher levels of bacterial killing ability between August and October compared with May through July. In addition, the high levels of baseline bacterial killing observed August through October may be sufficient. For the same reasons, energy is likely not prioritized toward the court response, explaining why we saw no change throughout the acute stressor. Across sexes, other traits within the individual may also influence responses. For my future work, I hope to complete my radioimmunoassays, conduct reproductive assessments, include morphological data in my statistical analyses, and add additional field seasons to my data set. I would like to thank my undergraduate field technicians, my mentor, Dr. Lori Neuman-Lee, and my various funding sources, including Arkansas State University. I'd also like to thank you for listening to my presentation today.